meeting is officially being recorded. Okay, so just a reminder, we did talk about mutations, so let's go over the two major types of mutations. We have two major types of mutations, point mutations and frameshift mutations. Point mutations are those that change a nucleotide, basically exchange one for another, and you can see it's called the substitution. And remember that if you just substitute one nucleotide, you're not taking one out or adding one, just subbing one for another. That will, because that only basically just changes one of the nucleotides in a series of 3, that just affects one codon, all right? And remember, all these changes are taking place in DNA. Although they could take place in RNA as well, it's significantly, mostly we talk about this in DNA. And then a frameshift mutation is when instead of a nucleotide being substituted one for another, instead what happens either is a nucleotide is inserted into the DNA or a nucleotide is removed. So because when you have a sequence of 3, if you add one, that makes it 4. So we would have a sequence of 3, and then every codon after that would be affected. Or if you delete one, you go from 3 to 2, which means you start reading the first nucleotide from the subsequent codon as part of that, right? So what frameshift mutations do, which is again insertions or deletions, what they do is they affect the entire codon sequence. So not just one codon, the whole codon sequence. So we talked about with point mutations, you can classify these in 3 ways. One, you can have where codon is basically, you do have a nucleotide exchange, but the resulting codon still codes for the same amino acid. So this is due to the repetitive nature of that codon table of the genetic code, or the degenerate genetic code, and the fact that we have multiple codons that code for the same amino acid. So this kind of overlap in that allows silent mutations to be ones that they do change the genotype, but the phenotype is not changed. The amino acid is the same. And then you can have a change in a nucleotide that would lead to a change in a codon and therefore an amino acid. And that if that change results in a different amino acid, which again is what we're looking at in this picture right here, where we go from GGC to AGC, which ends up changing our amino acid from glycine to serine, that is called a missense mutation. Think of it as like a mistake. So remember, silent, there's no change, silent. But missense, it's a mistake. You have a mistaken amino acid. The effect of this depends on the chemical properties of the amino acid. So like in that figure, if both glycine and serine were acidic, probably it really wouldn't affect your phenotype because the acidic amino acid would interact similarly to the previous one. If the chemical properties are vastly different, then that could potentially affect the phenotype. So it just depends on what amino acid was changed. And then finally, you can have what's called a nonsense mutation, and this is, you still get a change, so it's not a silent mutation, but instead of changing to an amino acid, it changes to a stop codon. So basically, in this case, translation would be terminated early. You'd get a shortened polypeptide. And that's shown, all of these are summarized in this picture right here. So you can just compare a silent mutation. Again, we go lysine to lysine, or I think that's in proline, but proline to proline because of that repetitive nature. Or we go from here, alanine to proline, which is changing the amino acids. Or here, we originally had a lysine. We had a change to a T, and that resulted in a stop codon. So notice that our overall polypeptide which was originally 7 amino acids long, is now only 4. So it's shortened. And then we talked about frameshift mutations, again, insertions and deletions. And you can see the big difference here is that not just one amino acid was changed, you can see all subsequent amino acids are changed. So a little bit more serious. We also talked a little bit about the fact that, you know, when we're talking about amino acid or polypeptide coding, we can also have genetic mutations take place in non-coding regions that would affect translation or transcription, sorry, either one really. And that can be if you have like a mutation in the promoter, that could mean that transcription was either increased or decreased, right? It could mean that maybe RNA polymerase can bind and go without an activator. You can have a mutation like that. That would increase the rate of transcription. Or maybe what would happen is you would have a mutation that would not allow an activator to bind appropriately, or transcription factor, and that would decrease transcription. So you can have a lot of effects on that entire promoter. Remember, the promoter region includes both the enhancer upstream and that core promoter, the TATA box, and the RNA polymerase binding site. So there's a lot of room to have mutations there. And then, of course, anything that would encode something that would regulate transcription or translation. You also have those intronic regions, what on this slide they call it intergenic regions. And those are less likely to affect gene transcription because they're not immediately coding. However, it's believed they do play a significant role in gene regulation. So less likely to affect gene expression, but that's not across the board. Okay, so you'll notice that on Tuesday I announced I added some things, uploaded a newer version of the genetics lecture. This is one of the slides I added because I thought it was interesting and relevant. I thought, why not talk about some effects that are noticeable from some of these genetic mutations? So the first thing I want to mention about genetic mutations is that they are extremely complex. So if you remember, when it comes to going from a gene to a protein, the vast majority of our body is not, isn't a result of proteins that are coded for by one gene, right? You have genes that can encode multiple proteins because of alternative splicing and all sorts of crazy regulatory things. Temporal or time-wise genetic expression can really affect the phenotype at any given time. So I mention this because your phenotype right now is not a result of that kind of classical view of like one gene equals one protein. It's all of those regulatory elements that are involved as well. So it's really important to think about when we talk about genetic mutations, it's very, 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 very rare to have a mutation, like to say that one mutation is what is causing a genetic disorder. So you can see over here in the top right, I have a little highlighted box that says that realistically genetic disorders are extremely complex. So you look up something like, you'll notice that I have disorders that involve point mutations. And then I have disorders that involve frameshift mutations. And you'll see that there is a lot of overlap. So cancer, Tay-Sachs is listed twice. I would be willing to bet that every single one of these could be listed in both columns. But the reason they can be listed in both is because obviously we've kind of talked about cancer already, but cancer is so complex. And as you know, cancer is when a cell starts replicating repeatedly. So we talked about those checkpoints, and when a cell kind of bypasses those checkpoints for mitosis, that's rule number one, right? So if it can bypass a checkpoint, then it can start replicating uncontrollably. So that would be maybe one mutation, is a mutation like a point mutation that allows it to start bypassing those checkpoints and start replicating. But realistically, most of our cells actually, you probably have a cell doing that every day, but there's a lot of other checkpoints that exist. Basically, our body has a way of saying, okay, the cell has started replicating, it's time for it to die. So a normal process called apoptosis, or apoptosis, however you want to say it, is programmed cell death. And that also has a ton of regulation involved with it. And so in order for cancer to evolve, you actually have to accumulate quite a bit of mutations. You have to mutate to allow the cell to replicate repeatedly, so bypass those checkpoints. And you also have to have mutations that basically stop the cell from dying. And so that's called, we talked about it previously, an immortal cell. For a cell to live forever, it doesn't have to just replicate, but it also has to make sure that it's not killed. So there are a lot of mutations involved in these disorders coming to be. Cancer involves a lot of point mutations, frameshift mutations. A lot of the cancers that tend to be more severe, like breast cancer as a result of the BRCA1 gene, and prostate cancer, those are the two off the top of my head. Those ones tend to have several more frameshift mutations. They're a little bit more severe. Those cancers tend to go fast. That's because of more drastic mutations. Some other diseases that result from specifically point mutations are, I guess I would say, have a high level of point mutations correlated with them or corresponding to them. Neurofibromatosis, sickle cell anemia. So sickle cell anemia, we talked about that already in our last unit when we talked about inheritance. So when your red blood cells go from big and plump to sickle shaped, kind of like a half moon shape, that's actually a point mutation that causes them to have that shape. Tay-Sachs disease, as well as color blindness. Frameshift mutations, cancer, again, some of the more serious ones. Not that any cancer is not serious, but I guess some of the more quickly progressing ones. Crohn's disease, cystic fibrosis is a frameshift, has a frameshift mutation associated with it, Tay-Sachs. Hypertrophic cardiomyopathy, which is actually a really scary thing. That's a kind of random frameshift mutation that occurs that causes the heart to kind of hypertrophy, which means kind of like grow. But basically what happens is this can lead to like cardiac failure, just like random cardiac failure. That put me on a terrifying rabbit hole is what that term did. Sort of an interesting thing is that some of you may have heard that there are some people that have a natural resistance to some infectious diseases. So most notably, we know that there are certain individuals in the population that are less susceptible to HIV. And those individuals actually have a mutation in a gene that codes for a cell surface protein called CCR5. This is an important signaling molecule for immunity, but it also is a receptor that's utilized by the virus to gain entry to cells. So if your body doesn't have this, you may have some like lessened, maybe slightly lessened immunity to certain things, usually like mild bacterial infections, but you would have resistance to HIV, which that sounds, pretty good. So that's kind of, not all mutations are necessarily like bad. So one thing I want to mention about these is that some of these, when I mentioned that these are very complex, they involve a lot of mutations, not just these. A kind of interesting thing is that not all of these mutations are necessarily causative. So for example, with breast cancer, the BRCA1 mutation is not what directly causes the cancer. It certainly contributes, but like I said, there's a lot of other mutations that have to take place for that cancer to progress. However, as you know, people get screened for the BRCA1 mutation, and the reason that is although that mutation is not solely causative for the cancer, it actually can be diagnostic. So some of these mutations are not necessarily directly or solely responsible or causative for these disorders, but they can be diagnostic, used for screening purposes, and also predictive, which is useful. Okay, so cool. Let's move on. I'm gonna show you a clicker question because I think that they're good practice, but we're not gonna spend a lot of time on it because I want to, move through genetics, guys, come on. This question, sickle cell anemia, occurs when a point mutation results in the change of one amino acid in the code for hemoglobin. So one amino acid is changed. So this causes the hemoglobin to clump and the red blood cells to sickle. What type of mutation is this? So it gives the kind of hint is that we do have one amino acid changed to a different one. So that tells us that it's B, right? So if that amino acid was changed to a stop codon, it would be a nonsense mutation. If the amino acid was not changed despite its mutation, that would be a silent mutation. If the entire reading frame or amino acid sequence was changed, that would be a frame shift, possibly a result of insertion. All right. Okay, so I had an excellent question last week. Let me pull it up. The question, I had two really good questions actually. So one of the questions that this slide specifically is going to answer is basically, the question was, how many cells with mutated DNA making the wrong proteins does it take to alter the phenotype? Or is it just one mutated genome? And the answer is, it depends on when and where the mutation takes place. So in this slide, what I want to show you is, again, where and when that genetic mutation takes place really matters as to the overall kind of effect of a genetic mutation. So remember that we have two types of cells in the body. We have cells that are zygotes, or sperm and an egg, that, that form, sorry, we have gametes, and then these lead to the formation of an embryo. I don't know what I'm talking about, but I haven't had enough coffee today, you guys. But you have cells that are what we characterize as germline cells. And what that means is that these contribute to the formation of an embryo. So again, this is going to be like sperm and egg contributing to an embryo. But those cells are going to lead, again, because of differentiation, to all of the cells in the body. So all of the cells in this body will result from the combination of these two cells. That's a germline cell. They are like the stem cells, right? Embryonic stem cells become all of the cells in the body. So germline mutations can be a lot more severe. And if they are a certain level of severity, usually that means that they do not lead to a fully viable fetus. And so a lot of times you may see people with genetic disorders, but those are what we call survivable genetic disorders, even if they were germline mutations. But a lot of the germline mutations are not survivable, which is one of the reasons that we do actually see a very high rate of miscarriage. And that's because genetic, germline mutations are not exceedingly uncommon. So with germline mutations, again, these are cells. The germline is cells that give rise to the cells that will become the entire organism, differentiate into all of the organism. So what this means is that if you have a mutation, let's say in, we see that the mutation is in the sperm cell here. That's why it's red. So the red is associated with the mutation. So you see a mutation in the sperm. In the embryo, it's the combination of the sperm and the egg that leads to the formation of the embryo. This means that it kind of depends on which chromosomes this fetus gets, but obviously it's going to get any of its Y chromosomes from the sperm, but also it's going to get half of its other chromosomes from them too. So what happens is that any mutations that were carried by that sperm also will differentiate and contribute to all of the cells in the adult organism. So all of the adult organism will carry that. As well as that, the cells that contribute to their production of the gametes will also contribute to that mutation. But you'll notice that only half of those will carry the mutation because half of the chromosomes are coming from the mother, which didn't contribute the mutation. So in the case of a germline mutation, that mutation can be pretty drastic and would affect the overall phenotype of the organism because every cell in the body would carry that mutation. So I have a question. Does the egg screen sperm for mutations when a sperm is trying to enter the egg? No, that's a great question. And the answer is no. It's really, really random. Actually, what happens is usually, gosh, I can't remember. I need to look into it, but I think I was asking my husband about this the other day, actually, because I was just curious. I think it is just one sperm and egg, but as for what sperm ends up entering the egg, there's not a lot of sperm that actually reach the egg, one. But then if you have multiple sperm that reach the egg, it's sort of like random chance which one is able to enter first. So the egg is not necessarily, there is no choice. It's sort of just like which one gets through. But let me look into that to see if there's, but I will say that the egg doesn't have the capability of screening, but the structure of the sperm matters. If it has to have like the correct shaped head to get in, basically. Good question. Yeah. So germline mutations, if they take place in kind of embryonic tissue or pre-embryonic tissue, like the germline, like sperm and an egg, those will affect the entire organism. However, you know that we also have non-germline cells. We have our somatic cells, or our diploid cells, and we can also have mutations occur in those. So this one, the effect of a somatic mutation really, really, really has a lot to do as to when, through development, that mutation takes place. So if it's relatively early, so we have a fetus develop, so we have non-mutated sperm and an egg develop an embryo normally, but that embryo actually has something causes, this is looking like radiation or something, causes some sort of mutation. You can see that mutation occurs in a clump of cells right here. And then that embryo keeps differentiating into a fully formed organism. It would be that area of cells, or whatever that area of cells will lead to, that has the mutation. So you can see sort of like a patch of affected area. So because at the point of an embryo, right, there's regions of the embryo that will become arms and legs and everything. You can see that in the embryo, it's sort of the region that will develop into the shoulder and the arm that receives that mutation. So it's sort of the shoulder and the arm area that have a mutation. So Could a mutation occur as an adult? Excellent question. So getting to another point. So that's a mutation occurring in an embryo affecting development, but absolutely yes. You also have mutations that occur as an adult. For example, a couple of them I can think of off the top of my head, skin cancer, right? So as adults, we're out in the sun and we have, exposure to UV light causes a mutation in our skin, which can lead to the formation of melanoma, right? That is a genetic mutation that leads to a phenotype, which is cancer. Another example of development as adults would be, a lot of the examples I can think of are like cancers, but there are more examples as well. But absolutely, yes, you can incur mutations as you become an adult. A lot of those are associated with cancers, though. And the reason that is because cancers start with one cell. So as adults, we are fully differentiated. And so any of the mutations that would occur would be in our differentiated cells, like our skin cells or our ovarian cells or wherever kind of associated cancers are. So excellent questions. So we can have a genetic mutation kind of anytime, but its overall effect really depends on what type of cell it occurs in. Is it a germline cell or is it a somatic cell? And also timing. So for example, like on the right, if we have a somatic cell mutation during the embryonic development stage, you can see that that affects those differentiated tissues. However, if that person is an adult without a mutation, and let's say you get like a little melanoma, typically they start out as the size of a freckle. So they're a very small area. So it just depends on kind of when in development those mutations occur, really impacts severity. All right, cool. So I always get a couple of questions about like what causes genetic mutations. There are a lot of things that can cause, unfortunately, there are a lot of things that can cause genetic mutations. You can have things that cause genetic mutations kind of spontaneously, which is what this table is talking about. So we didn't really go into transposons, but what I can talk about is the ones that we did talk about. So for example, the first thing listed is error in DNA replication. So when you're copying DNA, and let's say your DNA polymerase actually pops in the wrong nucleotide when it's copying it. So when you have DNA copied incorrectly, this is actually like very rare because DNA polymerase has really good proofreading machinery, and also your body has a whole repair mechanism for detecting these and changing these, which I'll tell you about in a minute. You can have some toxic byproducts of normal cellular processes. So starting today, we're going to talk about metabolism. Some of those metabolic products are called, of normal cellular respiration, is hydrogen peroxide, which we actually talked about in lab. We talked about peroxide produced by plants like turnips. And we talked about the fact that that's normally produced by, the plant creating ATP. Our bodies also create hydrogen peroxide as a byproduct of cellular respiration generating ATP. It's actually a toxic byproduct that can cause changes in DNA. So we have to get rid of it. We do have enzymes to get rid of it. Plants have peroxidase. We have an enzyme called catalase, and you can see it work. So if you ever, I'm sure several of you have like got a cut before, and if you get a cut and you put hydrogen peroxide on that cut, you know how it gets a lot of bubbles? So it gets bubbles because hydrogen peroxide is H2O2, and what peroxidase does, so you're pouring hydrogen peroxide on it, and your cells and everything in all your cells have catalase, which serves to neutralize hydrogen peroxide. And it neutralizes it by taking H2O2 and splitting it into H2O and O2, water and oxygen, which together are bubbles. So one thing I'll mention is a lot of the bacteria on your skin also have catalase. So hydrogen peroxide itself is not like an excellent antiseptic because it's automatically neutralized by not only your skin, but also the bacteria there. It does kind of bubble the dirt out and stuff like that, clean it. It's not necessarily doing anything to the bacteria. But I mentioned that just because you can like recognize what your body does to hydrogen peroxide. When hydrogen peroxide is created through cellular respiration, you have catalase to neutralize it into water and oxygen. That occurs throughout your entire body, not just on your skin. We can also have changes in nucleotide structure. So we're not going to talk too much about this, but a really common thing is changes in nucleotide structure can be like, you can get like what are called thymine dimers, whereas you have two thymine molecules and they'll kind of just like randomly dimerize or stick together. And transposons are really interesting because we're not going to talk about them, but what they are is they're little segments of DNA that have the capability of like splicing themselves out and jumping to a different region of DNA. So they have their own splice machinery. So they literally like are in one region of DNA and splice out and enter into another. Typically these jump around in introns, and so they're not really noticeable, but very, very rarely they will insert into a gene, which is kind of scary. All right. Interestingly, we're not sure if retroviruses like HIV either evolved from transposons or transposons are a remnant of the fact that we evolved from retroviruses. Something to think about, something to chew on. All right. And then, so those are just spontaneous things that just like kind of happen in the body that could cause mutations. You can also have environmental factors lead to chemical mutations. We typically classify environmental factors into two categories, chemical agents and physical agents. Chemical agents can be anything like cigarette smoke is an example of a chemical that causes mutations. That can obviously change not only tissue structure, which we talked about earlier in the semester, but structure of DNA, benzopyrene or BPA, and what was another one? I always forget. I'll think about it, but there's, I mean, the one thing to avoid here, and this sort of drives me crazy, is that you'll hear every day, it's like, broccoli causes cancer, and then, you know, like breathing causes cancer, or staring at a cell phone will cause cancer. It's sort of like, you get a lot of mixed messages, and a lot of these things, so cigarette smoke and BPA and a lot of these things, what happens, like cigarette smoke has a lot of chemicals in it that are directly related to seeing DNA changes. Other things, and it's like chemical substances, there are several chemical substances that can kind of lead to genetic changes, but like we talked about, a lot of these things have a lot of contributing sources for cancer to develop, if that makes sense. But the things listed here, BPA, cigarette smoke, have been, you know, directly linked. And then physical agents, so everybody knows about UV light. Certain X-rays can damage, that's a miscommunication. X-rays are known to lead to DNA change, but I mentioned that, so that would be like, and also like certain types of radiation can lead to DNA change. I cannot spell today. Too much. Okay, but I mentioned that because there is something called irradiation that they do to food. So I mentioned that because like x-rays can lead to DNA change. That's why when you go get an x-ray, like the x-ray tech wears all the equipment and they don't like your doctor really shouldn't unless there's a really important reason to recommend getting x-rays. And that, you know, radiation can cause DNA change. But I mentioned this because this is a common misconception that kind of like drives me crazy, is that irradiation, which is a way that we can treat food, irradiation is where we take food and we put really like low level radiation waves at it. And what it does is it actually kills bacteria on the surface of the food. And since bacteria mediate degradation of food, if you kill the bacteria on the food, that means the food won't degrade as fast. So this is really important because if you irradiate the food, you kill the bacteria, the food is left intact and fine. And if you then take that food and eat it, irradiation doesn't like contaminate your food. So you can eat that food and it doesn't have bacteria, it doesn't have any like radiation associated with it. It's not radiation, it's irradiation. And you can eat that and it will not change your DNA. I hope that makes sense. It's very important right now, especially because there's a lot of craziness with food accessibility and there's a lot of fear around irradiated food, but irradiated food is perfectly safe to eat. It's not the same thing as if you were to like hit food with radiation. It's a very different process. All right. What would a microwave be considered? So those, I was asking, it's funny, it's like I was having these conversations with my husband just yesterday. But microwave, there are microwave, like literally it's called microwaves. But those, let me look into that. Because microwaves are such small waves that they induce heat, but that's it, right? They only heat stuff up. The waves themselves don't necessarily do anything, if that makes sense, which is the difference than like X-rays or radiation, which those actually affect the DNA, the organism. So microwaves are a little bit different, but let me look that up. That's a good question. I don't know enough about that. I should have probably paid way more attention in physics, but I hated it. Not that that's an excuse. Okay, so we've talked a lot about how DNA can be damaged. But now what I want to talk about is when it's damaged, which again is an inevitability. We are going to get DNA damage. DNA damage is going to happen. How do we fix it? Because I will tell you again, like I mentioned, you are experiencing genetic mutations every single day, but we are not seeing the effect of those mutations every single day. So why not? And it's just because the body is like really good at repairing itself. So all organisms have the capability of genetic repair, especially those that are, well, I will say, yeah, all organisms. Viruses don't have this capability, which is one of the reasons they mutate so readily. But all living organisms made of DNA have the ability to repair their DNA because by nature, DNA polymerase itself, which performs replication, can very, very well proofread itself and correct itself. So that's good. If it ends up adding an incorrect nucleotide, it can actually detect that and replace it. So that's what's called, well, that's just the proofreading machinery of DNA polymerase. But you also have specific systems in every cell that are directly related to DNA repair. So you can see in this table, this is talking about like the three most common, not most common, three common, and I guess I'll say also like best understood mechanisms of DNA repair. You can have what's called direct repair, where you have literally an enzyme that's dedicated to repairing either incorrectly structured, so the wrong nucleotide, or broken DNA. You can have specific machinery that basically it'll see that like there is, if it sees like the wrong nucleotide, base excision means it's cut out, and then it'll repair that removal. So for example, if a nucleotide was inserted and causing a frameshift mutation, it will remove that and it just won't insert another nucleotide. And that'll make the reading frame correct. But if it was a substitution, it'll remove that wrong nucleotide and it will insert the correct one. And it uses a complementary strand of DNA to say like, is this right or wrong? And then also it can do what's called mismatch repair. So it's basically what happens is it'll look at both strands of DNA, and instead of like looking, like this table says, it's very similar to excision repair, but instead of directly recognizing like via a sequence that it's wrong, it's looking at the complementary strand and saying like, these don't match, right? So it'll be like an A and a G because A binds to T. So if it sees an A and a G, that's a mismatch. So it will correct that. So obviously it has to know which one belongs. So which one is the mismatch? And that's how refined it is. It's able to read and determine which one is correct. So I'm not going to spend a whole lot of time on this, but I do want to mention that this is just sort of showing what that second one, nucleotide excision repair, sort of looks like when it happens. So you can see in red, we have our DNA double-stranded, and see down in this DNA, we have one of those things I mentioned earlier, which is a defect in the DNA where two thymines next to each other just like stuck together. So again, that's called a thymine dimer. And that's actually a pretty common issue that occurs in DNA, thymine dimers. It can be very serious. So you see this thymine dimer. What happens is you have the nucleotide excision repair machinery. You don't need to remember these terms like UVRA and UVRB or ABA. All I want you to know that is this is the machinery, the NER machinery. So it's just kind of checking the DNA. It just kind of goes in and checks it. And what it sees is it sees a problem. It sees that thymine dimer, detects it. And what happens is it recruits some other machinery, as you can see over here, to basically, you can see it cuts out like a significant amount of DNA. You can see it's like, well, here's our mistake. We're just gonna like cut out everything around it and start from scratch. So what it does is it cuts out the entire strand of DNA where that defect is noted. So you'll have a big gap in DNA, and then it has another DNA polymerase basically come in, fill in the gaps, and correct. So that's why it's called nucleotide excision, because excision means to cut out. It cuts out that nucleotide and also all the surrounding nucleotides, and then repairs it by patching it up with a new strand of DNA that's read off of this complementary strand. So kind of crazy thing about this, again, this is present in all of your cells. All of your cells can do this. Very common process. It's always, you do replication and your body, double checks your DNA to make sure for mistakes. So one of the kind of crazy things is that I'm sure you've heard like HIV retroviruses actually insert into your DNA. So it's a common misconception that all viruses insert into your DNA. That is not true. Only retroviruses can insert into your DNA. Those are by far not the most common viruses that exist. But HIV is an example of a retrovirus. There's a couple others, but HIV is the most notable one. And HIV can actually, what it does is it infects cells. It is made of RNA, so it's called a retrovirus because it does transcription and replication. Retro means reverse. So instead of going from DNA to RNA, it starts as RNA and does reverse transcription, retro, back to from RNA to DNA. And then it takes its DNA and it inserts it into your DNA strand. And it does this because what it does is it takes its DNA and it makes little cuts in your DNA. And what happens is those little cuts trigger your DNA repair mechanism to basically say like, oh, there's cuts here. I need to patch it up. And instead of patching it up with DNA, it patches it up with the virus DNA. So that's how retroviruses like HIV actually become a part of their host genome, because they use nucleotide excision repair mechanisms to patch themselves into the actual DNA. And then every time that cell goes through replication or transcription, the virus's machinery also is created. Nice. Okay. Oh, I had a question. Good. Does it read the DNA the same way a ribosome would read it? So remember that this is DNA. So do you mean directionally? It would still read. So remember that this is either replication or transcription. So we're not going to have a ribosome involved anywhere here. So this would be complementary base pairing. So again, like with this strand, like if we had an A here, a T would be put here. If we had a C here, a G. So it would basically be complementary nucleotides. For directionality, unfortunately, you'll notice, luckily, this is a little bit different DNA polymerase, but typically it still does move. It still does move 5 to 3, or create 5 to 3. So directionally, yes. So yeah, very similar to still how all of the other machinery works. Yeah, primarily during replication is what we're talking about. Good question. All right, so that's our nucleotide excision repair. We have a lot to thank for that. Sure, very grateful for that. Okay, so notice there are terms and there are questions for genetics here. So go through those. Use these to kind of guide maybe something if you want to put it in the discussion board. What I'm going to try to do today is if you can get your questions into the discussion board, I'm going to go through last week's and this week's discussion boards and kind of aggregate questions. And based on those questions, I'm going to record a little kind of blurb and post it tomorrow. And you can view it whenever you want to, but it's basically just going to address some of the questions that you put on the discussion board. So if you could post your questions today, that would be ideal. If you don't get them posted by today, not a huge deal. There's already a ton of really good questions on the board. But yeah, I'll also do that for the Unit 3 exam one as well, which I'll post on Sunday. Okay. Let's move on. Yeah, that sort of finished in a timely manner. Just love genetics. So cool. All right, but let's move on to something also cool. Let's move on to energy and metabolism. Yay! Very important, very exciting. Yes, yes, yes. Okay, so a student sent me this last semester and I loved it. The day mitochondria went from being the powerhouse of the cell, which remember is not, membranes are actually the powerhouse of the cell, but the day the mitochondria went from the powerhouse of the cell to ATP synthesis by oxidative phosphorylation was a horrible day. And I'm here to tell you that Tuesday will probably be that day, but we're going to start it on this lecture today. We're going to work on it. We're going to get there. Okay, so let's talk about energy and metabolism. Everything in this lecture is coming from chapters 6 and 7. So next week you will have a Learn Smart over this as well. I actually might publish that Learn Smart a little bit early so you can just get a head start on it since we're starting it, but it won't be due until next Sunday. So you'll have plenty of time to do it. All right. Let's get in on it. So some of the objectives, what are we covering? We will talk about enzymes. We've already talked a little bit about enzymes in lab. If you remember those days when we used to have lab, we're going to revisit those concepts. We'll talk about some of the basic steps of cellular respiration and photosynthesis, two of the core metabolic pathways. All right, so why does this matter? Well, we're talking about all living organisms in this class, like the basis of all living organisms. And if you remember thinking back to like our very first week of classes, we talked about those principles of life. And one of those principles of life was principle 2, which states that all living organisms use energy. All living organisms use energy. And that's because cellular processes require energy. We've talked about transcription, translation. You learned a little bit about nucleotide, excision, repair. We've talked about replication. Every single little thing that a cell does, a big thing, you know, they're cells, so they're little. But every single thing that a cell does requires energy. You cannot do anything without energy. So, just a reminder, we can categorize any process in a cell as part of a cell's metabolism. So I'm reiterating this because, again, it is very common for people to say, what is metabolism? And for the answer to be cellular respiration, but that is not true. Transcription is metabolism. Translation is metabolism because they're all chemical reactions. Every single one of those is a chemical reaction in a cell. So any cellular process that involves a chemical reaction, which is all of them, is a part of the cell's metabolism. And we can divide those metabolic processes into 2 broad categories. We can divide it into anabolism, which is when we are building things, and we can divide it into catabolism, which is when we are breaking stuff down. So again, these are any pathway. So we talked about replication, taking a strand of DNA and making a new strand of DNA. That's an anabolic process, right? So anytime we're building something, that's an anabolic process. Primarily, so far, we've only talked about anabolic processes because we've talked about taking DNA and making more DNA, taking DNA and making mRNA, taking mRNA and making protein. That's all building stuff. But today, we're going to focus, and next week, we're going to focus primarily on breaking stuff down. Because for anabolic processes, we have to use energy to build stuff. For catabolic processes, that's how we get the energy that we can use to build stuff. We get energy from breaking stuff down because energy is stored in chemical bonds. Yeah. So we're going to talk about these, revisit these concepts. I do want to just mention that in sort of an ecosystem, you do have these, even though we take metabolism and separate it into anabolism and catabolism, these are intimately linked and depend on each other. So widely, cellular respiration, which is a process where we take glucose or sugar and turn it into energy, and release energy basically, is performed or is considered a catabolic process. So it kind of shows some organisms that only do cellular respiration and they take in sugar and then they produce energy. So as a byproduct of that energy, as we will learn, they also produce water and carbon dioxide, which are used by organisms that perform photosynthesis, which is typically considered overall an anabolic process. Because what you do is you take energy, you use energy, use some other stuff, carbon dioxide, water, sunlight, and you build sugar. So photosynthesis builds those sugars. And then that sugar is used by cellular respiration to make energy, and the cycle continues. I don't really love this slide because, as we will talk about, these organisms also do cellular respiration. So even though these organisms can also do both of these processes, so this entire cycle could be seen in this tree, these organisms cannot generate sugar. Sure. Okay. Anyway, let's not go there. But overall, we can categorize organisms into producers and consumers. Consumers are the ones that kind of utilize, like, eating plants for nutrients, using those nutrients to kind of create energy. They produce byproducts that are used by these plants to create energy. And it's this beautiful cycle of consumers and producers in an ecosystem. It's a lot more complex than that. So I don't really like this slide, but okay. All right, so let's get to the basics of it. So what are we talking about when we're talking about energy? I say you need energy to do this, but what the heck is energy? Well, energy can be classified a variety of ways. So first off, it's important to note that at its basis, energy is kind of grouped into two categories: kinetic energy and potential energy. Kinetic energy is the energy of motion. So you can think of this as we talked about UV light. UV light is waves, X-rays are waves, microwaves are waves. All of those things are forms of energy, and that's energy of motion. Thermal energy, heat. The reason thermal energy is heat is because when atoms are heated up, they start moving really fast. That's why they are kinetic energy, the energy of motion. Alternatively, you can have what's called potential energy, and potential energy is stored energy. So an example of this is chemical energy, which is probably, we're going to be talking a lot about chemical energy in this lecture. So I have a picture in the top right over here of a molecule. I want to say it's like methane. And we have, for example, carbon. The reason I think it's methane is I think that this is a carbon and this is 4 hydrogens. But we have a molecule and we have a bond created between two atoms in that molecule. And that bonds in general form, and when they form, we put energy into forming bonds, right? It takes energy to create a bond. But when bonds are broken, energy is released, right? So when a bond is formed, we put energy into the bond forming. If we have a bond intact, that is potential energy in the form of chemical energy, because it's a chemical bond. It's a bond between two atoms in a molecule. So chemical energy, which is the energy stored in bonds, so potential energy, what we can do is when we break that bond, energy is released. So breaking that bond and having that energy released, once it's released from that bond, it's kinetic energy. So we're kind of going to be in this lecture with all of these metabolic processes. We have to work within the laws of thermodynamics. So there's two laws of thermodynamics that are applicable to this lecture. First off, just know that the law, thermodynamics is the study of energy transformations. So basically, when I talked about having that stored energy in a chemical bond, breaking that bond and releasing it, that transformation, transforming that energy from stored energy to, or potential energy, to released energy or kinetic energy, that's transforming it. That is a thermodynamic reaction. So that's thermodynamics, studying those energy transformations. There's two laws here that we will be working within. The first law of thermodynamics says that energy is constant. It cannot be created or destroyed. And this is very important, and I'm really bad at this. So like correct me in the chat box, like when I do this, because I will do this. We cannot create energy. Energy exists. There is a certain constant of energy in the universe. But what we can do is we can store energy or release energy. So whenever we need to use energy for a chemical reaction, we basically have to release energy in order to use it. If we want to store energy in sugar, we have to store it, right? So we're not creating it, we're not, you know, like necessarily like ever getting rid of it, we're just transforming it from one state to another. There is one thing that happens though, is that specifically with every transformation of energy, and this should mostly be from the transfer, I think this should kind of be specified that if you, I'm going to write this on the slide, anytime we have a transformation of energy from potential energy to kinetic energy, heat is released. So I'll show you a picture of this in a minute. Sorry. Whoa. Will allergies ever go away? Okay. All right, so with every transformation of energy, heat is released. So this means a little bit is that every transformation of energy, even though there's a constant amount of energy in the universe, sometimes those transformations are not 100% efficient. So whenever you move from potential to kinetic energy, you're also releasing heat. Heat, as you see, is a form of kinetic energy. It is a type of energy. It's just not for metabolic purposes, usable energy. So whenever you go from stored energy in a chemical bond and you break that bond, you release some usable energy, but you also release some heat, which is not necessarily usable energy in metabolism, in metabolic processes. It can help though, as we'll talk about. All right. So this is kind of just looking at those transformative processes, and I actually love this slide because the bottom panel is looking at cellular respiration, which is one of the core metabolic processes in a cell that gives us energy, and then comparing it to the transformation of energy that we see associated with a car converting gas into, well, like movement. But you've probably noticed, so right, gasoline is it's a hydrocarbon molecule, meaning that you have a huge line of carbons attached to hydrogens. And then you also have some oxygen going into this reaction. But a car takes fuel, gasoline, and puts it through the engine to turn it into movement. But you'll notice when you're driving a car, when you get out of the car after you've gone somewhere, if you touch the hood of the car, you'll notice that there's a significant amount of heat released. Like the car is hot, and not because we're in Arizona. The car is hot because it's been converting gasoline into energy. And what is happening is some of that energy is becoming usable energy going into moving the car, but some of that energy is being released as kinetic energy in the form of heat. Some of the byproducts of that reaction are carbon dioxide and water, which is why sometimes also when you stop the car, you'll see some water dripping out of the exhaust pipe. Hopefully not too much. All right, and then on the bottom, you see a cell, and you can see that the fuel for a cell is actually very similar to the fuel for a car. It is also a hydrocarbon molecule, but it also has some oxygen attached to it. And it is glucose, C6H2O6, H12O6, C12H. Why can I not remember this? C6, H12, O6. You guys, I have not had enough coffee today. How can I not remember this? Let me double check on that though. Pretty sure I'm right though. Okay, we have glucose, which has carbon and which has hydrogen, makes it a hydrocarbon base, but you also do have those oxygens attached to it. It's sugar, really good form of fuel. It also requires oxygen. But what that cell does is it takes in that energy source, and very similarly to how a car uses that energy, and turns it into usable energy, which is energy for cellular work, which is ATP. But via these processes, we also have heat released, which you can detect because whenever you exercise, your cells are using sugar to turn it into ATP for all that cellular work, for building new cells, for creating more ATP, for turning on genes that, you know, need to create muscle fibers, right? Your cells are working, but you can feel that because when you exercise, what happens? You get hot, your body gets hot. And that's because your cells, as they're going through these processes, as they're doing these metabolic reactions, they're also releasing heat. You'll notice the waste products are exactly the same, which is, I think, really cool. So we're going to be looking at this process in depth, but the reason that's important is just to know that this is not 100% efficient of a process, those transformations of energy. And that's just because, again, even though we're always transforming energy from potential to kinetic or kinetic to potential, every time, with every transformation of energy, mostly we talk about it in terms of transformation from potential to kinetic, there is some loss of usable energy in the form of heat. So that can be beneficial. Okay, so whenever we talk about converting energy, it is chemical reactions that convert energy. So like we talked about, cellular respiration converts stored energy or chemical bonds into usable energy, which is ATP. Photosynthesis is also a huge chemical reaction that converts kinetic energy basically into stored energy, or kinetic to potential, in the form of sugar. And so we can actually classify chemical reactions as, we already talked about classifying metabolic reactions as catabolic or anabolic. We can also classify these based on kind of what they're doing with energy. And this is very similar as classifying them as catabolic or anabolic. We can classify them as either endergonic or exergonic. Ooh, did we see a big old mistake on this line? Produces, do we produce energy? No. Releases energy. And then we can say requires energy input. So an endergonic reaction means that we are taking energy and storing it, right? We're requiring energy being put into this reaction. So what that would mean is that this would be putting energy into the building of bonds. So this would be an anabolic. Reactions are typically your endergonic reactions. Whereas exergonic reactions are those that release energy. And when we release energy, if we are releasing them, that means they were previously stored. So if they were stored, they were potential, and if we're releasing them, they're being converted or transformed from potential to kinetic. So this would be a catabolic reaction. All right, so our endergonic reactions are those requiring energy input. So we have to put energy into the reaction. Again, this is typically, you can think about it as if we're contributing energy to something, we're contributing to it building a bond. And if we're building bonds, we're building molecules, so that's an anabolic reaction. On the flip side, we can have an exergonic reaction. those are those that release energy. So if we're releasing energy, that means it was originally stored. So chemical bonds, breaking, that's a catabolic reaction. Okay, so you can see here on the right what this means. This is kind of a weird way to show it, but I actually really like these figures. On the top, you're looking at an endergonic reaction. So here on the left, you have your reactants. So the chemical reaction, your reactants are whatever go into a chemical reaction. So let's say your reactants can be two molecules. We'll call them A plus B. Your two molecules are A and B. All right. And what we want to do is we want to take A and B and we want to build a bond between them. All right. So anytime we build a bond, we just model that by saying AB. We just shove those two molecules together. So again, anytime we go into a chemical reaction, we can write that. We can write that. Give me. As kind of this. You write your reactants and then you write your products. So I would say A plus B. Those are my two reactants. I want to put them through a chemical reaction, which is shown as an arrow, and then you show your product. So what the product of that reaction is. And if you shove them together like this, that means a bond was created between them. So in this case, with my endergonic reaction, my reactants individually, because they don't have any chemical bonds associated with them, have very low potential energy. So potential energy is on this axis, low potential energy. But what we do is we put energy in to those molecules and we form a bond between them. And what that means is that our product, which is AB, bound by bond, our product, because we formed a bond, there is more energy stored in that product. So you can see with an endergonic reaction, because we put energy into that reaction, we go from low potential energy to an increase in potential energy. But of course, that does require energy, which is what we're putting in here. So on the flip side, here, B, we're kind of doing the opposite. So we're taking a molecule that has relatively high potential energy, so meaning that it's going to have bonds in it. And what we're doing is we are trying to release that energy to use it for something else, right? So essentially break a bond. So down here, it's essentially the opposite, right? What we do is we take a molecule with high potential energy, meaning it has bonds, and we break those bonds, which releases energy. But if we're releasing energy from this molecule, right, we split it apart, we broke that bond, that means that those individual products have less energy than what was put into that reaction, the reactants. We release that energy. So you can see the amount of energy released. That's kind of the difference in the reactants and the products. So what this means is that in an endergonic reaction, the reactants, oh my gosh, have less, I'm sorry you guys, I need a new keyboard. Less potential energy than products. It's on the way. Hopefully Amazon considers it essential, because I for sure do. And an exergonic reaction, the reactants have more potential energy than products. All right. Hopefully that makes sense. So we're done, but I want to fill this in. So we're talking about chemical reactions and converting energy, right? Building bonds or breaking them. In the chat box, can you tell me, like, how do we do that? What molecules do we use to perform chemical reactions, or basically facilitate them taking place? Give you a hint, it's a type of protein. Anybody? Anybody? Yeah, thank you, Erin. Thank you, William. Yes, it's chemical reactions are mediated by enzymes. Woohoo! We love enzymes, right? So I mentioned that because you know what we're going to talk about on Tuesday? We're going to talk about enzymes. So we'll talk about the fact that how we get this energy put into this reaction, or how we get this energy released, is via enzymes. Okay, so we are out of time, but as usual, I've got a little time. So if you want to hang around, if you have some questions, feel free to ask them. If not, right now, and you think of one later, for sure put it in the discussion board. Even if you've posted already, I have no problem, you know, like, you have a minimum of one post and one reply, but if you want to post and reply more, great. Like I said, I'm going to be going through that, those discussion boards today, and hopefully I thought it might be a fun little thing for me to like do a little update based on them, just a little recording thing, and to kind of cover the stuff that seems to be confusion topics. I'll do that from last week and this week, and then yeah. So if you have questions now, I will also be available for office hours today. I'll have my room open. You could call that coffee hour if you just want to stop by and sip coffee electronically. That's cool too. But if you don't have questions, then feel free to peace out. I miss you guys. I hope everything is going well, and have a wonderful day. Thank you.